AS Level Biology 9700, and today's topic is cell structure. Students, this is the fifth video on this topic, and the today's subtopic is cilia. A cilium or cilia are small hair-like protuberances on the outside of eukaryotic cells. And students, what are eukaryotic cells? Which have proper membrane-bound nuclei, and they are primarily responsible for locomotion. Locomotion or locomotive, they act as locomotive organ because they help them to move from one place to the other. So either of the cell itself, so they locomotion not only for cell itself or the other fluids which are present on the cell surface. So they are also involved in mechanoreception. So mechanoreception is actually the ability to detect and respond to mechanical stimuli. So what are those mechanical stimuli? Let me write over here. Mechanical stimuli. What are those mechanical stimuli? They can be the touch, pressure, vibration, and sound. So they are the mechanical stimuli. So they are also involved in mechanoreception. So they can receive all these mechanical stimuli. There is even a class of microorganism named for these small structures. They are called as ciliates. So ciliates are the protozoans that possesses cilia, which they use for both locomotion and feeding. So the basic function or the main function of cilia is locomotion. And if we see its structure, so we can see the cilium is made up of microtubules, already discussed in the previous part. What are microtubules? They are made up of proteins. So these microtubules are uh, coated in, you can say, coated in plasma membrane. So the microtubules are small hollow rods made of protein tubulin, alpha tubulin and beta tubulin, already discussed. Each cilium contains 9 pair of microtubules. 9 pair of microtubules are there, and forming the outside ring. So they will be, so these pairs, they will make in this way. In this way, they will make up the ring and 2 central microtubules. So two central microtubules, so they will be 9 plus 2. And the structure is known as axoneme and the arrangement as 9 plus 2 and arrangement ubiquitous in motile cilia. So the microtubules are held together by cross linking proteins. Between the 9 outer pairs are motor proteins called dyneins. So you can see the structure of on the next slide. You can see the structure of these cilia. They are very much similar with the structure of microtubules and centrosomes. So these microtubules, they will held together to make the structure of the cilia. Here you can see the axoneme, which is made up of 9 pairs of microtubules, which make, you can say, the just like the very close to the boundary, and in 2 are present in the center. So they will make 9 plus 2. Motile cilia, they will have the 9 plus 2. Other, the primary cilium, they will not have this 2 which are present in the center. I would like to say the structure will be little bit different in both primary cilia or in motile cilia. Motile cilia will be those cilia which help in the movement or help in the locomotion. So they are very close to the structure of the centrioles. You can say, just like them, because centrioles are also made up of these microtubules. The next subtopic is microvilli. Microvilli Already you have listened this word villi, and the similar is villus, which are present in on the internal surface of small intestine. So microvilli are microscopic cellular micromembrane protrusions that increases the surface area for diffusion and minimize any increase in volume and are involved in wide variety of functions, including absorption and excretion. So, they are finger-like projections which are present on the internal surface of the small intestine, and their uh, main function is to increase the surface area for more absorption and for diffusion. So, in this way, they are involved in these major functions which are absorption and excretion. So, they are present uh, in the small intestine, and you can see its internal structure. Microvilli, they will have epithelial outer membrane. And inside this membrane, you can see the finger-like projection, and the finger-like projection, you can see the capillary blood vessels. So blood vessel is there, and the lacteal lymph will also there. It means the during this capillary, the food is during absorption, the glucose and amino acids are absorbed and uploaded in simple cell, and through lacteal you can see the lipids, glycerol plus fatty acids, they are uploaded through this lacteals. So in this way you can see the microvilli are very helpful inside the cells. The next subtopic which I'm going to discuss is the cell wall. A cell wall is an outer layer in case of plant cells. Not there, the cell wall is absent in animal cells, you know very well. So a cell wall is an outer layer surrounding certain cells that is outside of the cell membrane. The cell wall provides strength and structural support to the cell. It not only, you can say, outer layer, but it also provides the support and strength to the cells, and it can control to some extent what types and concentrations of molecules enter and leave the cell. Strength, protection, protection, shape maintenance. So it also control the term extent what types and concentrations of molecules enter and leave. Almost, you know that it is porous in nature. It has small pores inside, so the molecules can easily pass through it. But to some extent, we can say that it also controls some concentrations of molecules which can enter or leave the cell. The materials that can make up the cell wall differ depending on the type of organism. So, depending on the type of the organisms, uh, the different materials of cell wall: cellulose, lignin, peptidoglycan. So these are the different materials which can make up the cell wall of different organisms. So this is the reason that the different materials are involved in different types, because they different organisms have to perform the different functions. They belong to the different types. So that is the reason the different materials are involved in the formation of cell wall. The cell wall has evolved many different times among different groups of organisms. The cell wall has a few different functions already discussed in the previous slide. So it is flexible. It also gives the flexibility. You can say it gives, it supports, it protects the cell. So, but provides strength to the cell, which helps protect the cell against physical damage. So it also gives the cell its shape and allow the organisms to maintain a certain shape overall. Cells, uh, plant cells can maintain their shape because of this cell wall, whereas the animal in animals it is not present. So the animal cells can change their shape. They have different shapes. So the cell wall can also provide protection from pathogens such as bacteria. They are trying to invade the cell. So it also protects the cells from different microorganisms. So, or the pathogens. Pathogens are the disease causing agents. And very important point about the cell wall is the presence of plasmodesmata between the cell wall of two cells. So it means all cells are connected with one another. It happens only in plants. Keep in mind, it is happening only in plants. That is, the cells are connected with one another. So plasmodesmata, small channels. So these data are small channels or pores, you can say, which connect the cytoplasm of plant cells to each other, forming a living bridge between cells. You can see over here the living bridge. So this is the uh, here I would like to show it with a different color. This one. So this is the plasmodesmata. So plasmodesmata are the small channels which connect the cytoplasm of plant cells to each other, forming a living bridge. You can see the different water can move through these plasmodesmata. Each cell is not completely isolated by the cell wall. We can say that cells are not completely separated from one another. They are connected with one another with this plasmodesmata, and through this desmata, the water can pass through. And there are two pathways which are involved in the movement of the water or other solutions. You can say they are apoplastic pathway and the symplastic pathway. In apoplastic pathway, the water pass through the cell wall. It is not uh, passed through the cytoplasm. Cytoplasm is not involved. It means there is no role of plasmodesmata in this. But here you can see in symplastic pathway, the water can pass through the cytoplasm through these plasmodesmata. So in this way, the water can move from one cell to the other through these plasmodesmatas. Now the next subtopic is vacuole. A vacuole has a broad definition. Vacuoles are present in both animals and plants. But the difference in the both animals and plants is in animals, there are many small vacuoles present which have different names. Some of them are called as contractile vacuoles. Some of them are called as food vacuoles, contractile vacuoles, and some of them are called food vacuoles, etc., according to their function. But in case of plant cells, a single large vacuole is present in the center. A single So, vacuole has a broad definition, include a variety of membrane-bound cells. It is double membrane. It has double membrane structure around it, single, large in the center. So, the membranes are composed of phospholipids. The structure of this double membrane is the same, just like the plasma membrane. So, made up of phospholipids, but each organism may use slightly different phospholipid. It means the different organism will use the different type of phospholipid molecules. So embedded in the membranes are proteins. Keep in mind the structure, double well structure just like this, in this way. Double membrane, and embedded in them are the different types of proteins. Plasma membrane and different types of proteins, just like that. But the different type of phospholipids are there. So the function of these proteins, which are present in the membrane, transport molecules cross the membrane or give it structure. Various combinations of these proteins allow different molecules to handle and hold different materials. So these different types of proteins which are present, they will handle and hold the different types of materials to pass through the cell membrane, to pass through this vacuolar membrane, which is called as tonoplast. So the main uh, membrane of vacuole is called as tonoplast, just like the plasma membrane. So various combinations, it has the main function of the central vacuole is to maintain turgor pressure in the cell. Turgor pressure is the pressure of the cell's contents pushing against the cell wall. When the vacuole is filled with water, high water potential is there. At that time, the turgor pressure is produced, which will push the cell wall in the outward direction. So it is only found in cells that have cell walls, such as those of plants, fungi, and bacteria. You can see over here a single, this is the plant cell. So in the plant cell, you can see a vacuole is there. This is in the center, vacuole is there, and the membrane of the vacuole is called as tonoplast. So this is only the plant cell. If we talk about its outer membrane, which is tonoplast, so the term tonoplast is derived from the word origin. Tone means tension, stretching, or tone, and comb means molded. So it means it will be in the fold form. It will be the tonoplast is also called as vacuolar membrane. Already told you vacuolar membrane or tonoplast. So both are, and it is cytoplasmic membrane that surround vacuole, just like the plasma membrane. So thus separating the vacuolar contents from the cytoplasm of the cell as a membrane. So tonoplast is directly involved in the regulation of movements of ions around the cell and isolating any possible harmful material that is threatening to the cell. So as a membrane, so as a vacuolar membrane, tonoplast is directly involved in the regulation of the movement of ions and around the cell and isolation any possible harmful material that is threatening to the cell. So it will not allow those types of molecules to enter into the cell. So in this way, it also protecting the cell. So the tonoplast contains numerous proteins that are responsible in the transport of small solutes and salts, and proteins that are involved in the fusion of membrane and remodeling the allowed delivery of macromolecules by vesicle traffic. So in this way, you can see the role is same. The role of the tonoplast is same as the plasma membrane performing its role, and the structure is also resembling with one another. Only there is difference of the phospholipid molecules. So the different type of phospholipid molecules can be there in the plasma membrane and in the tonoplast. And the function of vacuole is to bear the main function is to bear the, or you can say, uh, the turgor pressure, protection. It also store the different materials into it, and you can say the protection as well. So in this way, vacuum also play a very important role, and tonoplast help to play all these roles. So in this way, it was all about today's lesson, and wait for the last and the sixth video on this topic, cell structure. And please don't forget to like, share, and subscribe my channel, because in this way you will be able to get the notifications of the solved papers and the syllabus which we are going to cover during this summer. Thanks for watching.